All right, so while people are coming up, this is the session on new approaches to cancer treatment. But after discussion amongst us, we decided to also expand this to cancer prevention. So that's what you'll be hearing about this morning. So we'll have 5 talks. Each one of us will present our viewpoint on what we think is the best approach, the newest approaches to cancer treatment and prevention. We will hold questions until the end, where we'll have a panel discussion and audience Q&A. I will start first. My disclosures are here. So I'm a cancer immunologist. Similar to Jim Allison, we both study the evolution of immune responses, what's involved in generating an immune response against cancer, and we know that evolution can sometimes become stalled. So this slide shows you the type of pictures that we were puzzling about when I was a trainee in Steve Rosenberg's lab in the 1980s. We would look at microimages of tumors that were growing in patients, but that were heavily infiltrated with CD8 positive killer T cells. And if we took those cells out in the laboratory and under the right conditions with cytokine incubation, et cetera, we could see that those T cells actually could recognize that patient's cancer and could even destroy it on plastic. But somehow in the patient, the T cells just weren't working. They were stalled. And then, of course, in the 1990s and 2000s, you just heard from Jim about all the advances that were made in understanding why endogenous anti-tumor immunity can be stalled, but also how to reactivate it in a way that cancer cells can be killed. Next slide, please. And so with the development of immune checkpoint blockade, we have found, as you just heard from the previous speaker, that antibodies blocking anti-CTLA-4 and or anti-PD-1 can cause the regression of very advanced cancers that haven't responded to other forms of treatment. And this can happen across a wide range of tumor types. At my count, we're now up to 22 different kinds of cancers that have FDA-approved immune checkpoint inhibitors that can prolong life and even appear to cure some patients. So today I would like to tell you about work that we're doing that's sponsored by NFCR with a rare skin cancer called Merkel cell carcinoma, where anti-PD-1 therapy has really been transformative in the outlook for these patients who previously had no effective therapies once their disease had metastasized. So Merkel cell carcinoma affects about 3,000 new patients a year in the United States, and it's long been thought that there was an immune component to this cancer because the incidence is much higher in elderly patients and also in patients who are immune suppressed, such as patients who have had a solid organ transplant and are then on immune suppressive drugs. Interestingly, most Merkel cell carcinomas are caused by a polyoma virus. And as I mentioned, before the advent of anti-PD-1, the outlook was very dismal for patients with advanced disease with average survival of less than 1 year. Next slide, please. So I mentioned there are these 2 different subtypes of Merkel cell carcinoma, and genetically they are at the extremes of tumor mutational burden for what we know about human cancers. So the virus-positive Merkel cells have an extremely low tumor mutational burden on the order of just a dozen mutations in the entire tumor genome. So really the virus is driving those cancers and not probably the very few mutations that are present. On the other hand, the virus-negative Merkel cell carcinomas are caused by a carcinogen, ultraviolet light, and they have an exceedingly high tumor mutational burden. So we also knew from research at Hopkins that many Merkel cell carcinomas express PD-L1, the ligand for PD-1. And Paul Niem's group in Seattle had also shown that T cells that recognize Merkel cell carcinomas in patients express PD-1 and TIM3 and the other checkpoints that you just heard about. And so we formed a collaboration and drew in other medical centers to test the idea that PD-1 blockade could be effective in Merkel cell carcinoma. Next slide, please. Next, please. And this slide shows you the results of a clinical trial that actually led to FDA approval for anti-PD-1 in 2018 for this disease. So the response rate was very high. Over 50% of patients in the trial responded. And on the left there, what you see is a waterfall plot. And so each bar is an individual patient, and the bar is dropping below the minus 30% line, or what we call a response in oncology. So they're color-coded in red or blue for the virus-positive or virus-negative tumors. So you get the idea that we saw about the same rate of response whether the cancer was virus-positive or virus-negative. And on the right is a spider plot showing you how quickly those responses occurred. Of all the human cancers that have been treated with checkpoint blockade, Merkel cell is among the quickest in terms of the kinetics of response. And it also shows you that the responses are very durable. They go on for many months, and now with extended follow-up, we know for years. So this information shows us that these Merkel cell polyomavirus antigens can actually serve as tumor rejection antigens. Next slide, please. And what that means, thank you. What that means on the right is that we now have an easy way to monitor anti-tumor immunity in these patients by monitoring responses against the Merkel cell polyomavirus, which produces T antigens, a relatively small number of amino acids. You can make all the peptides, you can make tetramers to find these T cells in the blood or in the tumor. And this is much easier than doing what I'm showing on the left there, which is figuring out for each patient what the cancer-associated mutations are, and then trying to track each of those mutations in individual patients. Is very time-consuming and labor-intensive. Next slide, please. So our NFCR project is a collaboration between Johns Hopkins and Paul Niem's group at University of Washington in Seattle. Our goals are to increase our understanding of anti-tumor immunity and anti-PD-1 response in Merkel cell carcinoma by looking at the transcriptional programs on a single cell level of tumor-specific T cells. And we want to compare the virus-specific T cells to tumor mutation-specific T cells. We also want to compare the virus-specific T cells to T cells that are specific for other kinds of viruses like influenza, EBV, or CMV. Okay. Okay, so to fast forward this, we took information that had been generated in the Smith and Pardall labs at Hopkins from lung cancers that had been resected after neoadjuvant anti-PD-1 therapy. And they had published a group of genes that was associated with mutation-specific T cells in lung cancer. So the first question we asked was, could we use that same gene signature to find a Merkel cell polyomavirus-specific T cells? And so what you're seeing, there's only one group of green dots that's rising above the rest, and those are the T cells found in the tumor that are specific for Merkel cell polyomavirus. So the T cells in the blood specific for the same virus did not have that transcriptional profile. And now in our project, we have already studied 3 patients with Merkel cell carcinoma, looking at T cells infiltrating their cancers, and compared them to the transcriptional profiles that were found by Smith and Pardol and published in 2021 in Nature. And very interestingly, the Merkel cell virus-specific T cells resemble the mutation-specific T cells from lung cancer. And so they are characterized by a larger group of genes, but I'm showing the top 3 that we think are driving this transcriptional profile, which is low IL-7 receptor, high CXCL13, and high CD39. This is very preliminary information, but it suggests that by identifying these genes, we may be able to hone in on targets that are going to be useful when combined with immune checkpoint blockade, and potentially avoid some of the relapses that do occur in about 25% of these Merkel cell patients over time. And so here I have acknowledgments of the research group. So today then, we're going to hear from 4 panelists, Nellie Polyak from the Dana-Farber Cancer Institute, Avery Posey from University of Pennsylvania, Azra Raza from Columbia, and Thea Tilsty from UCSF. And without further ado, I'll ask Nellie to come up.
Okay, good morning. Thank you for giving me the opportunity to present our work funded by NFCR. And if I can get the first slide. I don't know if this is working. Oh, they actually have to open, yes. So the story I tell you about brain metastasis and how the special environment in the brain particularly makes breast cancers resistant to treatment. These are my disclosures. And, okay. So my lab studies breast cancer, looking at breast tumor evolution. And as you know, breast cancer is not a single disease. There are 3 major clinical subtypes of breast cancer that are characterized by the expression of the hormone receptors, ER, PR, estrogen receptor. I don't know if this is working, the pointer. So hormone receptors, that's characterized the hormone receptor positive disease. HER2 positive, HER2 is an oncogene expressed on the surface of the cells. It's about 15–20% of the tumors, breast tumors have this. And there is a triple negative disease that is characterized by basically triple negative because it's lacked the estrogen, progesterone, receptors and HER2. And these subtypes are very relevant in terms of they have different risk factors for development. They also have different approaches for treatment, and then the outcome and the metastatic progression also different according to the subtype. So today I will tell you about a story in HER2 positive breast cancer. So this is one of the breast cancer types that has expression of this oncogene called HER2 on the surface of the protein on the cells, cancer cells. And there are various ways to treat these tumors. One of them is that there are antibodies against the HER2 protein itself, such as trastuzumab and pertuzumab. There are more recent years antibody drug conjugates developed like TDXD and TDM1. That's very exciting and have significant improvement in treatment. These are basically antibody against HER2 coupled to a drug. And then there are also small molecule inhibitors that either inhibit the kinase, tucatinib, neratinib are examples for that, and then chemotherapy is used as well in combination with the other agents. So my lab has been studying the resistance to HER2 targeted therapies quite extensively, looking at the heterogeneity. That's one of the mechanisms of resistance. But today what I will tell you about brain metastasis. And brain metastasis is one of the most devastating progression of the disease. In breast cancer, brain metastasis is the second most common brain metastasis of all cancer patients. Lung cancer is another one that's very commonly develops metastatic disease. And unfortunately, as you see on this survival plot, patients who develop brain metastasis has a shorter survival, even among all the metastatic patients. And because we are improving the treatment of HER2-positive disease in the periphery, patients actually living longer, but then most of them develop brain metastases. So in HER2-positive and triple-negative disease, almost 30% of the patients, or even 50% more recent years, will eventually relapse with brain metastasis, and even ER-positive disease, it's 15%. And we're talking about total breast cancer is 240,000 women a year. So 30% of that, that's quite large number, and this is only about brain metastatic disease. So, let's see. When we started this project, what we thought about is what's special about the brain. And as you know, the brain is a very unique organ, has many different metabolic state and cellular composition, and we have multiple projects in the lab investigating this. In this particular project, we wanted to see how the microenvironment in a brain, particularly the stromal cells, could potentially influence resistance to treatment. And then what we are hoping that with better understanding these interactions, we can actually improve treatment of the patients. Next slide. This shows a model that we developed. One more click. Yes. I just show you the schematics. So basically developed a system where we can detect the cancer cells that are interacting with a stroma or not. Basically, the red color indicates the cancer cells. The green one is the brain metastatic stroma. And then when the cancer cells interact with these green metastatic stroma cells, they turn on a gene that's blue, and then they basically turn purple, because red and blue together is purple. So that's the system we used. And I don't know if it's, okay. So first I show you that when we co-culture the brain metastatic stromal cells with the cancer cells. So this kind of shows the schematic. The red is the monoculture. When you have the green co-culture, that's the co-culture. And on the plot, what it shows that we tested multiple different HER2 positive breast cancer cells. And then on the top is also many different brain metastatic stroma cells. And then we also tested different types of HER2-targeted therapy, like neratinib, lapatinib are small molecule inhibitors, but also TDXD, which is one of the antibody-drug conjugates. And then we used 5-FU as one of the chemotherapeutic agents. And the blue color shows that basically those cells are surviving and they're not responding to treatment. So as you saw, like many of the stromal interactions basically makes cancer cells resistant to this HER2-targeted therapy, regardless of what it is, whether it's an antibody or small molecule inhibitor. Then we wanted to investigate what is happening in the cancer cells when they interact with the stroma. And we did multiple different profiling. This one is looking at gene expression profiles. So we basically sorted the cancer cells that interacted or did not interact with the stroma. And we did expression profiling. On the panel, this is a volcano plot. Depicts the genes that are upregulated or downregulated in a cancer cells when they interact with the stroma. And one of the top ones there that I like to highlight is MUC2, very highly upregulated. MUC2 is a glycoprotein expressed on the surface of the cancer cells. And then on the left panel here, you see again that if we're looking at these glycoprotein signatures, so it's not only MUC2, a single gene, but also the glycoprotein signatures seems to be upregulated when the cancer cells interact with the stroma and they respond to treatment. And this is, of course, in our experimental model, but in patients also, the metastatic lesions have higher expression of these glycoprotein signatures. What are these glycoproteins? This is basically sugar coating on the cells. So our cells, most cells in the body, and particularly cancer cells, have this glycocalyx, which are basically proteins that have some sugar on top of them. And this is very important for for many processes in the body. It's also important for protecting the cells from pathogens like viruses or bacteria, and also regulating the recognition of the cells by the immune system. But at the same time, unfortunately, it's also contributing to resistance to treatment because the sugar coating can prevent the entry of the drugs, and particularly in the HER2-targeted therapy, is blocking the binding of the antibody to the surface. And then we wanted to actually show on this plot that when we co-culturing the stroma with the brain metastatic stroma with the cancer cells, we see upregulation of MAC2 and other glycoproteins. And I don't know how well this projects, but again, the red is the cancer cell, green is the stroma. And if you see those halo kind of around the cancer cells forming when they interact, that is the sugar coating. So the sugar coating seems to be upregulated by the interaction with the stroma. And then in collaboration with the Bartosi lab, we actually did global glycoprofiling on the cells and on the resistant cells, because we're particularly interested in the ones that are not responding to treatment. And as you see, the yellow color shows like upregulation of particular glycoproteins in the resistant cells. And what looks interesting on this plot, that the cells resistant to the small molecule inhibitors have a very different profile from the ones that are resistant to antibody drug conjugates like TDXD or TDM1. We just got this data fairly recently. We're very excited about following this up. A lot of interesting findings. And we actually investigated what is the whether we can overcome the resistance by treating the cells with an enzyme called mucinase, which can remove the sugar coating. And this plot here shows that when we interact with the stroma and they don't respond, but when we're treating them with this mucinase enzyme, then we can decrease the survival and basically respond, and the cancer cells respond better to treatment. On the right-hand side, this is a Western blot looking at the signaling downstream of HER2, and I just like to highlight a point here, like when we have a co-culture with the stroma, this phospho-EGFR and phospho-HER2 is not decreased as effectively by the drug, but when we're treating with a mucinase, then we are able to block signaling in the tumors. On this one, next. Yeah, next slide. We actually also showed that if we overexpress this MAC1 protein, then the cancers become resistant to treatment. So as you see here, these are the parental cells, overexpression, they really change morphology. They are not responding to treatment. The green one is the overexpression. They have a constitutive upregulation of the signaling pathway. And on the next slide, if I could, yeah. So we also injected them into mice, and then they form brain metastases, these MAC1 overexpressing cells, much more efficiently, and they are not responding to treatment. So these are the control cells. They have brain metastases, and then they respond to neratinib treatment, as you see here. This blue color is the metastatic lesion. It goes down. But when we're overexpressing, basically they have a sugar coating, then they're not responding to treatment as well. And that just shows the quantification. So just to summarize what I show you very quickly, what we see that the stromal interaction in the brain makes the cancer cells resistant to treatment. Some of its resistance is coming from the upregulation of the glycoproteins and glycocalyx. The why this is important, we want to investigate how we could block this interaction, and these upregulated glycoproteins at the same time, they could actually represent new targets, like new antibodies, and CAR T cells also could develop against these and improving treatment of the patients. And the last slide is just an acknowledgement. Oops. Acknowledgement of the people who did all the work. Marianne, Laura are 2 postdocs in the lab. Caroline de Pignon was an undergrad in the lab. And then Val Weaver is our collaborator, and Caroline Bertozzi at Stanford, who have been collaborating on the glycoprotein profiling as well. So thank you for your attention, and we greatly appreciate the support of NFCR.
Thank you for having me here. My name is Avery Posey. I'm a PhD geneticist that was converted to cancer immunologist, and I focus on the incorporation of what we think or know to be required of the immune system to treat cancer into a gene therapy for T cells to generate synthetic immunity. And this is a therapeutic called CAR T cells. And in this project, I have a clinical collaborator who's an MD, Dr. Courtney Hutchin, who's in the back of the room at the Oklahoma University. And so it gives me access as a PhD to an investigator who has access to clinical samples for similar disease interests. Next slide. These are my disclosures. Next slide. Next slide. So in the development of CAR T-cell treatment, if you follow me in this diagram at the top of the figure and work clockwise, T-cells, a white type of white blood cell, are removed from the peripheral blood of patients. And then they're genetically engineered in the lab to incorporate the genetic material that encodes for these new molecules. They're grown in a laboratory and then reinfused to a patient for treatment. Next slide, or next. And this is data from the first patient treated at the University of Pennsylvania, now more than 12 years ago. What you'll see in the figure to the right is a large population of CD19 positive B cells in this patient. This patient had chronic lymphocytic leukemia. Next. And 1 month after treatment, in the bottom figure, you'll see that there's no more of those cells remaining. So that shows the potency of this type of therapeutic. That patient actually remained in remission for over 10 years with a population of circulating CAR T cells in the peripheral blood. And we think that is a clinical correlate that's going to be really important for treatment of many cancers, especially solid tumors. Next slide. And the way in which we build these molecules is by incorporating pieces of immunological receptors, immune receptors, together to form a new receptor that retargets the T cell towards a particular target antigen. Next. And in this figure, you'll see that we incorporate that into a lentiviral vector, transduce T cells. That now brings on a new receptor. Next. That then allows the T cell to target, to treat or to kill target expressing cells. Next. But one of the limitations of this type of therapeutic are the molecules that we're targeting. Most of the antibodies that we have in clinical use to target cancer target tumor-associated proteins. These are proteins that may be overexpressed on cancer, but they're found at low levels on normal tissue. Next. And this has led to toxicities that we've observed. Next. Next. Including against multiple antigens, HER2, using the antibody we just heard about, trastuzumab as the binding domains for the CAR T cell, carbonic anhydrase 9, mesothelin, which has been used in the treatment of pancreatic cancer in the past, and PSMA most recently for prostate cancer. Next. So the molecule that we're interested in is CEACAM6 for this project. CEACAM6 is known to have biological functions that contribute to enhanced tumor growth, including inhibiting the immune system through interactions with CEACAM1 on T cells, but also contributing to enhanced survival of tumor cells. Next. And it's known to be overexpressed in many cancers, many solid tumors, including lung, pancreas, stomach, colon. And you can see immunohistochemistry below of tumors from multiple patients with pancreatic cancers, staying with an antibody called MAB109 that binds to CEACAM6, and low level of expression in normal pancreas, but also low level expression in other healthy tissues. And next. And we know that this antibody actually has a requirement for the glycosylation of CEACAM6. So if you remove the N-glycans on total protein lysate with an enzyme called PNGASE-F, MAB109 can no longer bind to CEACAM6, whereas other CEACAM6 specific antibodies that don't require glycosylation can. Next slide. And this type of targeting of glycoproteins is something that my lab is really adept at. So we were interested in targeting changes in post-translational modifications on tumor-associated proteins. And our first candidate was actually MUC1. And we used an antibody that, that is distinct from glycan-specific antibodies or peptide-specific antibodies. These antibodies, like the one in the middle here called 5E5, targets glycopeptides as a complex antigen. So the antibody has a requirement both for the glycan and the protein. And if you look at the videos to the right, you'll see immunofluorescence, co-immunofluorescence showing in green staining of this type of complex antigen on healthy tissue as well as tumors. And in tumors, you find this brightly expressed on the surface of the cell, and whereas in healthy tissue, you find it in the Golgi where O-glycosylation is processing, but never at the surface of the cell. Next. And so for targeting of CCAM6, or this N-glycan of CCAM6 on pancreatic cancer cells, we evaluated a panel of CARs that all differ based on their co-stimulatory domain, because that contributes to the effector functions as well as persistence of these CAR T cells. And initially we show just evaluation of these cells, these CARs all expressed on the surface. That's important. That's not given for every CAR molecule that we generate. And we compare this to anti-mesothelin CAR T cells, which, as I mentioned, have been tested previously in patients with mesothelioma and pancreatic cancer and ovarian cancer. Next. And initially, when we take fresh CAR T cells and acutely stimulate them with tumor cells, they all work. They all kill. All of our MAB109 CCAM6 specific CARs kill, and perhaps better against some tumor cell lines than the anti-mesothelin CAR, which you'll see to the right. Next. But where we start to find differences in these CAR molecules is in the cocktail of cytokines that they produce after stimulation with tumor cells, where there's a difference in the quantity as well as then the type of cytokines they produce. Next. Oh, sorry, thank you. And I showed you that killing data with acute stimulation, but what happens when cells are within a tumor and they're chronically stimulated by an antigen and they have to continue to work and work and work? Just like us, they get tired, they get exhausted, as much as what Jim Allison was talking about earlier. And so we test this in vitro with acute stimulation, and then we expose cells to a tumor for 1 week, constantly replenishing the cells, and then ask them to kill, or 3 weeks. And what you can observe is that one of these CARs, those co-stimulated by CD28, consistently loses activity after a chronic stimulation. We think that this is related to T-cell exhaustion, and there's lots of molecular work that back these assays. Next. We've evaluated these CARs in multiple tumor models, preclinical tumor models, using pancreatic cancer cell lines. The first one here is BXPC3. Actually, both I'll show you will be BXPC3. But here we're evaluating a difference in the design of the molecule, in which we make a change in the orientation of the antibody, MAB109, that we're using to target cells. And in this case, we found that both designs work to control tumor growth, preventing it from what you see in the control group. These are mice treated with T cells that aren't targeting the tumor. And you see that our CARs are effective at targeting and killing the tumor. But actually, that's very important, but that's not the only correlate that we look for. Next slide. And in this most recent model, we see that when we evaluate the differences between the signaling of these CAR molecules, that one of them stands out as both being better in terms of anti-tumor efficacy, as well as having lasting persistence of these cells. So of the CAR T-cell products that are regulatory approved in the world, there are 9 to date, 6 in the US and 3 in other countries. 7 of them use the co-stimulatory domain 41BB that is known to have low potency but high persistence. And 2 are co-stimulated by CD28, which has high potency and low persistence. And our data, as well as in other models that we perform, support these clinical findings. But we find that CD2 co-stimulated CAR T cells, which are not commercially approved, but have been clinically tested in our TNMUC1 CAR T cells that I showed you earlier, show increased efficacy beyond what we observed for 41BB, but they also show increased persistence beyond what we can see for CD28. And so we think that this is a CAR design that may be efficacious for this target antigen as well as others in the treatment of pancreatic cancer. And next slide. So I've told you today about this panel of CChem6 CARs we've evaluated for their different functions in terms of co-stimulatory domains. And we find that CD2 seems to be an optimal co-stimulatory domain for the design of these CARs for pancreatic cancer. And in work with my clinical collaborator, we've tested these CAR T cells against patient-derived organoids and are now moving to test these in patient-derived xenograft models with the hope of eventually moving these into the clinic. Thank you.
Let me bring us back to the reason that we are all collected in this room today. I'll present to you the case of Andrew. In 2016, he developed tingling in his arm. He's taken to the emergency room where he becomes quadriplegic within hours. Neurosurgeons could only remove 90% of the 9-centimeter large glioblastoma multiforme, the deadliest brain cancer known to man. So from this moment on, every one of his treating oncologists knew that his prognosis is hopeless. But what follows? Surgery, chemo, radiation, immune therapy, more surgery, more chemo, more radiation. He suffered unbearable toxicities. The tumors continued to grow, and he died within 16 months of diagnosis. This poor boy was 22 years old, and he was the best friend of my daughter, Scheherazade. How many nights did I get a call from Scheherazade saying, "Mommy, you're supposed to help cancer patients. You're not helping Andrew. This boy has been in and out of my house since he's a kid." How helpless did I feel in front of his mother, her only son, dying now. How badly we are failing our patients. It's not just Andrew. Look at the age-adjusted mortality. I can't make this up. Yes, there is a 1% decline, but that is mainly from anti-smoking and anti-obesity campaigns. I came to this country as a 24-year-old fresh medical graduate, started to treat acute myeloid leukemia in 1977 with 2 drugs we popularly call 7 and 3. 7 days of Aracy, 3 days of donomycin. Today, in 2023, I'm using the same exact 2 drugs, 7 and 3. It is a complete embarrassment. What are the results? In 1977, 5-year survival for AML was 26%. Today, in 2023, it is 28.5%. Glioblastoma, we are not doing well. We are not doing well with most of them. And this is why a cry of lamentation from me is maybe we are doing something wrong. Stop chasing after the last cancer cell and maybe try to find the first cancer cell. Look for the initiation. Well, the problem is, how do we find the first cell? Because by the time cancer is diagnosed, there are hundreds of millions of cells already. Well, one possibility is that let's see if we can produce stress in the lab and what happens to the cells. So we used multiple ways of producing this stress, and we observed a very strange thing, appearance of giant cells, wild-type cells growing happily. Once we stress them, giant cells appear. And we caught them on camera showing polyploidy, and sometimes up to 32N DNA. What are these cells? We followed one of these cells. Nothing happens for 3 days. At 73 hours, it suddenly starts giving birth to babies. It's like a somatic cell pregnancy. And so we have caught this over and over on time lapse. These giant cells appear. They start giving birth to 2N DNA cell, one giant cell giving birth to so many. After a few weeks, all you see are the small cells. That is the cancer you diagnose. But we have missed the entire initiation story of the first cell by looking at only fully established cancers. Can we isolate these cells by size? So in 2015, I set up a filtration device in my lab at Columbia University, and literally using a coffee filter technology, because these giant cells have been identified in primary tumors, but they're very rare in the primary tumor. Now that we are doing liquid biopsies, we can catch them from the blood because we enrich for them. And so when I looked at patients, just 10 cc's of blood passing through a filter, we started to find these giant cells. Compare it to the size of a leukemia blast under it. That's a blast because it has a nucleolus. You see the giant cell is pregnant with like 20, 30 cells in it. And now we have published this data on hundreds and hundreds of patients with MDS and AML. I'm not talking about tissue culture cell line. I'm not talking about mice. These are cells that have been isolated from human leukemias, patients. Every time you see these bizarre giant cells with multiple small cells. In solid tumors, compare the size of the giant cell to the white blood cell. In fact, in 5,000 tumors, every single one was positive. Just looking at 7.5 ml of blood. More interesting was, once you give your standard of care treatment for cancer, and post-therapy, you take blood from cancer patients and look for giant cells. If you don't find any, survival is a straight line. Even if you find a single one, there is a relapse. And so we know these giant cells are important. My hypothesis is that under stress, two cells which are stressed fuse together, because fusion is a very normal response to stress. You double the genome instantly. You don't need to go through mutations or anything. And then the nucleus keeps dividing, the cell doesn't divide. With time, depolyploidization occurs, and that's where you have the actual cancer. We come in with treatment, we only get rid of the cancer. The giant cell survives. And that is what, in time, is going to cause recurrence. So can we imagine a two-step approach in which we first give standard of care to kill the cancer as best as we can right now, and then come in with treatment and elimination of the giant cell? Well, what about future patients? Can we prevent by taking out the giant cell early? With that in mind, I created the oncology think tank 5 years ago. Participants were from the major institutions in this country and some of our industry partners. And we published a paper, an opinion paper in Scientific American. Conclusion of the entire oncology think tank was, we have to find cancer before stage 1, because even stage 1 requires slash, poison, and burn. These are horrible, horrible treatments that we should not be giving to anybody. And so the only way is to find it even earlier. And how do we find that? Well, we proposed that identify a group of individuals who are at high risk of developing cancer. Do you know that 1 in 5 new cancers appear in a cancer survivor? My own husband, Harvey Preisler, who got his first cancer at 34 years of age, survives it. At 57, in the prime of his life, he's taken by a leukemia that was completely unrelated to his first cancer, and he dies. 1 in 5 is a large number. 20% cancers are diagnosed in a cancer survivor. Just do the math. If there's 1.9 million cases, this year 400,000 plus will appear in a cancer survivor. We can collect. These cancer survivors are coming to the centers all the time for follow-up. We can take non-invasive samples from them, try to see if we can catch the first cell early, and what about studying the rest of the samples for multi-omics technology, all the things that you're hearing about today? Well, with these ideas, I went to the acting director of the NCI, Dr. Doug Louie, who is here, and he was extremely supportive. He gave us a lot of his time, and because of his encouragement, I was able to do a pilot study at Columbia University of the first 500 cancer survivors, and we are finding amazing, amazing results with it. And so my idea was, well, we can now extend it to the rest of the institutions who are poised to start collecting samples. Within 2 years, we'll have enough samples, and 50,000 cancer survivors, a few thousand will have developed a second cancer. We have their samples from before and after. I really urge all of you, you are the leaders in oncology today. I stand in front of you as a failed oncologist who's very embarrassed to see 30 to 40 cancer patients every week and not having any better news to give. Please, let's try to rethink what we are doing and try to study patients and human samples. I will not show you this last thing because I've run out of time, but thank you.
We just heard from the previous speaker that we have to do better, and we can do better. So our approach to trying to address the questions that she just posed is a refined approach of studying the emergence and the control of solid malignancies very early in their process. If we can understand why and how these cancers emerge, it'll help us clinically. So we study human disease during its natural progression from healthy cells all the way to malignancy. There's no interruption with chemotherapy, radiation, etc. And we focus on early intervention so that we can avoid the morbidity, the mortality that Dr. Raza just described to us. Most of the field takes an epithelial-centric view of cancer progression. There are mutations in cancer, in a cell, they accumulate, it forms cancer. We have a slightly different view of this. We believe that the cancer is not only the epithelial cells, but also the stroma. And of course, many people believe this as well. And we believe that it's the endothelial cells, the fibroblasts, the immune cells, all interact together with the epithelial cells to cause this emergent entity to come out, as well as the ECM proteins, extracellular matrix proteins, that are destroyed and created as it goes from normal to malignant and the left to the right. Now, one of the reasons that we came to this realization was from work that we had done a few years ago. And what we did, the question we asked was, if we take some cells from the stroma of a cancer and allow them to talk to normal cells, what would happen? Would they have any effect on cancer? And so what you're seeing here is work done with human prostate tissue taken from patients. And if we put the normal epithelial cells, mix them with a little bit of the normal fibroblasts in Matrigel, and slip them under the renal capsule of a mouse where it feeds it vascularly, what we get is the recapitulation of a normal tissue. But if we take these red cells to the right, which we called carcinoma-associated fibroblasts, and allow them to talk to the normal prostatic cells, epithelial cells, what we got was a pre-cancer. It looked exactly like PIN, prostatic intraepithelial neoplasia, and if we did this with breast, it looked exactly like ductal carcinoma in situ, a pre-cancer, DCIS. Interestingly, if we took those healthy epithelial cells from the prostate and simply immortalized them, so they're not tumorigenic yet, they're simply immortalized, and now we allowed those red calf cells to talk to them, what we obtained was something that was 500 times bigger than the first thing that I told you about. If you cut it open and look at it, you can see that it's cancer. And so the yellow arrow indicates the signaling that came from these calf cells, carcinoma-associated fibroblasts, that increase proliferation, angiogenesis, mutagenesis, and reduces apoptosis and adhesion, as you can see in the histology. This was published quite a few years ago. In more recent papers, what we've done is we've looked at other aspects of the stroma, other cells, and here's just a piece of information with looking at pericytes from the stroma of cancer, and you can see, I hope, that the signals that are sent by the pericytes increase the proliferation of epithelial cells, tumor cells, by over tenfold. They deposit much, much more collagen and fibronectin. And interestingly, they deposit much less laminin. And the reason that this is so interesting is because on the top you can see that that amount of laminin, if you layer the epithelial cells on that matrix, you have proper polarization and differentiation of these epithelial cells. But with the reduced amount of laminin, neither of those occur. You've disrupted polarization and differentiation. So what I've told you right now is that stromal signaling is dominant in determining the malignant phenotypes of the epithelial cells. But what I also want to tell you is that these changes in the stroma occur early. This is not a change in the stroma that is induced by the tumor. It actually precedes the formation of the tumor. And so this early proactive event, it's a coordinated stromal response. I showed you fibroblasts and endothelial cells, but I could show you many more interactions. This coordinated stromal program is activated early, before the tumor arises, and it can be activated by wounding, radiation, high mammographic density, chronic inflammation, all things that are known to create people at high risk for cancer. Now, the work that I just showed you has been done in vitro, it's been done in vivo, and if any of this makes any sense, we should be able to make predictions from these data and take it to the human condition. And so one of the things that we asked is, are these cells that show a pro-tumorigenic program, are they present in human beings in precancer or before, and can they predict who's going to go forward to cancer? So what I'm showing you here is a retrospective cohort that was provided by Karla Kerlikowski, and all of these bars indicate ductal carcinoma in situ biopsies. We have 35 years of follow-up for this, and what I'm going to show you is the dots at the top are the ones that progressed to invasive cancer. Many people know that with DCIS, only 1 out of 10 patients which are diagnosed with this precancer actually go on to invasive cancer. What we found is the ones that go on to invasive cancer are the ones that have this pro-tumorigenic program, as you can see with the red bars. The ones that have the anti-tumorigenic program, the green and the red, the green and the blue, have a very low probability of going forward. And so now people that are diagnosed with DCIS have some place to go, some assay to look to see if they are high probability of progressing or low probability. And this gave us encouragement about the approaches that we were taking. So many people have been studying stromal contributions to facilitate the progression of malignancy and to control malignancy. We've heard beautiful talks this morning, mostly from the immune side of the story, and this is very intensely studied. But what I wanted to do is to remind you that the stromal components can also inhibit cancer, and in some cases they can even revert cancers, and this is underappreciated. And one of our earliest winners of the Snegogori Prize was Beatrice Mintz, and I wanted to remind you of this paper 50 years ago where she took a tumor cell and she put it in the blastocyst of a mouse, and what that mouse, the pup that emerged, contained genetic material from the tumor cell, but went forward and did not have cancer, was perfectly fertile, and went on to have other babies of its own. So we wanted to try and understand more about this side of the story, but of the many somatic cells in our body, only a few ever find themselves on a path to malignancy. So how can we go to the earliest cells that Dr. Raza just implored us to do? Well, one important clue is that nature has provided us is that injured tissues are often at high risk, and that's what we indicated before. And I wanted to draw your special attention to chronic inflammation, because when you have chronic inflammation in a tissue, it results in adaptive response, which is a metaplastic lesion. And you can see at the bottom, this telling you about gastrointestinal reflux. The acid goes up into the esophagus, and you can see that the squamous layer of the esophagus, after it's injured, the cells take on a new identity, which is gastrointestinal, and it's called metaplasia. The metaplastic lesion is directly linked to the incidence of cancer, and so it alerts us to when and where cancer actually starts, because it's the gastrointestinal metaplasia that gives rise to the dysplasia and the cancer. So now we know where to look. And so by combining all of these multi-omics analysis, single-cell RNA sequencing, ECM proteomics, spatial transcriptomics, as well as allowing the cells to talk to each other, as I indicated perfectly previously, under the auspices of the CRUK Grand Challenge, we mined the information to get info on risk stratification, prevention targets, and therapy targets. I'm going to show you finish with a sample. Here we're taking bronchial epithelial cells. We're growing them in co-culture. The epithelials on the top, the stromal cells on the bottom. This is air-liquid interface in hopes of generating the ciliated cells and the secretory cells that are so prevalent in the bronchial airway. And you can see in this air-liquid interface, there's a beautiful, fully differentiated functional tissue. You can even see the cilia from the side or from the top. That's what you're looking at there. They beat all in the correct direction and at the right rate that you see in the human being. We then wanted to ask, having established that, can we force this tissue to go down the progress to malignancy? So I showed you normal co-culture. This is what happens when you co-culture those same epithelial cells with a carcinoma-associated fibroblast. You see metaplastic transformation. You see squamous metaplasia. And we found that if we identified the pathways that were responsible for this and can inhibit them, we can actually prevent the metaplasia. Or if it's already established and we compromise those pathways, we can reverse it. And there's even epigenetic memory of what it had been. If we add more stress, we can get it to go on to dysplasia. And so we've been able to do this not only with bronchial airway, as I'm showing you in the middle, but also with the progress of esophageal cells to gastric metaplasia and beyond, and stomach cells all the way to malignancy. I want to finish with by saying that the studies that I just showed you CAFs from a squamous lung cancer that induced the malignant change. But if I take the CAFs from the esophageal cancer, or the gastric cancer, or a breast cancer, it induces the same metaplastic change. So that means we're looking at something that's very fundamental. Likewise, if we take the carcinoma, if we take the fibroblasts associated with the metaplasia or the dysplasia, the pre-cancers now, before the cancer has arrived, they will also induce this metaplastic change, telling us we're looking at a shared mechanism that occurs early. This now provides us with the information that we're not looking at just 1 or 2 or 3 or 4 cancers, but we're studying one fundamental process, and it allows us to act early and to target emergent interactions. So stromal reprogramming provides new directions to prevent and revert multiple cancers. This was done by a wonderful group of talented people, and as I said, it was done under the auspices of the Grand Challenge. Thank you very much.
In the interest of time, I think we'll condense the panel discussion a little bit, and if some of you have questions that we don't have time to answer here, maybe we can carry that over into the coffee break. But let me just start with one question, and this one's going to be for Thea and Nellie together. Okay, so you both mentioned, that tumors are complex ecosystems of interacting cell types. Cancer researchers, the people in this audience, have tended to focus on one cell type at a time, and everybody has their favorite cell type. Mine is the immune cell. But we now appreciate that there are other factors at play. So I'd like to hear from each of you. What do you consider to be the dominant cell or cells in the tumor microenvironment that are controlling carcinogenesis and cancer evolution? And what modern technologies have been most helpful to you in studying cell-cell interactions in the tumor? And finally, how do you think we can effectively interrupt pro-carcinogenic cell-cell interactions to move to the next step in therapy. So maybe I'll start with Nellie.
Okay, so as you know, we have been one of the first group that actually really emphasized the different cell types in the stroma, and we did a very comprehensive profiling almost 20 years ago when technology was not there. I don't know if I could say there is a single cell type that is critical, because what we see that these cells are interacting with each other and modify each other and recruit each other. So we like to view it as a whole, and I actually like to go beyond the cancer, because what we and others have seen that cancer is really a systemic disease. So there is a communication not only between cancer cells and an immediate microenvironment, but also the body. You know, the cancer patients have changes that we see that triggered by the tumor, and as you know, also the immune system is basically changing when you're developing cancer. So we like to look at the patient as a whole, not really just separating cell types. Of course, the immune cells are very critical, but there are many other cells in the fibroblast, and where we study in breast cancer, the myoepithelium is also very important, and the endothelial cells and the pericytes, as Tia was focusing. Technology advanced so much in the last 20 years, and especially in the last 10 years. Now we have so many methods for single cell profiling of, you showed some slides. We can basically isolate every single cell and determine the gene expression, the epigenetic, the mutation profiles, the proteomics now also. We're also doing some metabolomics. So we can do really high resolution of assessing every cell. This is on a dissociated tumors, but also on whole intact tissues. There are many, many technologies that lets you do spatial transcriptomics, basically looking at the expression, the proteins, and then the computational tools like artificial intelligence basically just look at, integrate all this data together and then give you predictions of, like even an H&E slide, which is routinely used. Using AI, you can make predictions of who can progress, but you know, it's not enough to predict who can progress. You need to understand why and how, and that's where dissecting these technologies comes in. And I think it's just amazing how much we can learn. And I'm, you know, also a big advocate of really studying the patient, because I always feel like the experiment has been done. We need to figure out what happened, and of course, we do experimental modeling as well, but the technological advances at the single cell level and the topology has really enabled us to dissect tumors at the level that we haven't seen before. Okay. So I can't give you an answer of what's the single cell type. I think it's all together.
Okay, thank you. So Thea, you've emphasized CAFs in your talk.
I'll be very brief.
Yeah.
I agree with what Nellie said completely. I think of the development of from normal to malignancy as a musical composition. And so you can't say that one is dominant over the other. Like a jazz ensemble, sometimes you have a drum solo, sometimes you have a trumpet solo, and the melody is thrown back and forth between, and the cells interact off of each other. The point I want to make is that as you go from normal to early precancers and late beyond, the biology actually changes. So who might be dominant early is not who's going to be dominant later. It goes back and forth. The last thing to answer your question about technology is I think a big breakthrough has been spatial omics. What can be done now spatially with genetics, RNA, proteins, etc., metabolomics, is that you see the cells intact, just like Dr. Razzo was saying. You see how they talk to each other, how far the conversation goes, and how they react to each other. So I think spatial omics is a huge step forward.
Thank you. Avery, you talked about adoptive cell therapy, and of course, this has had huge success in hematologic malignancies, but not so much in solid tumors. And so now you're applying your research to solid cancers. Why do you think it hasn't been successful to date in most solid cancers?
So I think there's several barriers to overcome. Antigen's one. There have been two different CAR T-cell products that have shown efficacy in glioblastoma and neuroblastoma. Those are GD2 targeted CAR T cells, a glycolipid, as well as in gastric cancers, which is Claudin 18.2 CAR T cells, which is an alternatively spliced isoform of a protein. And so I think that identifying the best target is one hurdle. Engineering your cells to last for a considerable amount of time to mount what will be a very suppressive and resistant microenvironment, as well as overcoming some of those additional hurdles in the microenvironment, resisting TGF-beta, the tumor stroma, as well as employing potential combinations to degrade some of that as well. So there's lots of challenges in this area.
Thank you. And Azra, you addressed cancer evolution from the first cell, and you study hematologic malignancies, which is a very nice model for those types of studies of cancer evolution. But can you tell us how successfully has that kind of thinking been applied to solid tumor evolution? What do you see are the parallels?
I honestly don't think that we know even the tip of the iceberg about what is happening in terms of, we know definitely that there's something called a convergent evolution. Because all cancers, whether they're liquid or solid, in the end have two things in common. Number one, they can ignore growth inhibitory signals. And number two, they do not mature. And this is common to liquid and solid tumors, which means that the starting point somewhere has to be the same. Whether that pro-inflammatory environment causing the stress and forcing some cells to develop a malignant phenotype as a response to stress is coming from pathogens or from autoimmune disease or from exposure to toxins, any of those possibilities clearly is giving birth to some environment, as some of you have been pointing out, which is forcing cells to misbehave. And cancer is nothing but a stress response which is exaggerated and gone awry. And so I feel that the evolution depends a lot upon the organ in which the cancer is, the host responses, the immune system, 1,000 other things. But in the end, the first cell, why aren't we even looking at that? Why are we constantly looking, trying to chase down the last cell? I just don't understand it, because really the key is what our grandparents always said: prevention should be the best cure.
Okay, that's a good thought to end on. I do want to give the audience a chance to, go ahead.
I just want
Thea.
to say
Yeah.
the first prize winner of the Sten Gregory Prize was Hal Dvorak. Cancer is a wound that will not heal.
Yeah,
He said it.
that's right. Yeah. Okay, burning questions from the audience? Please.
Hi, my name is Karen Fontana. I'm CEO of BioClips. I'm in a phase 1 trial, and if it's true that we have to move to the trial, to earlier, remember that our phase 1 trials are limited to refractory resistant patients. And our drugs, all of our drugs, are really probes for biology. But we're not allowed to use them in that way because we're limited at how to get into phase 1. We're limited where we can raise money after phase 1 if we don't see a specific, response in our first set of patients. And so how do we change the system to make something happen?
Can I answer that? So I think you're so spot on, and we do some phase 1 studies, and I think when we're asking a single drug to cure a cancer that resisted like 10 different lines of therapy, from evolutionary point of view, it's just set to fail. And so many drugs I think we're throwing out of the clinic that could work earlier step. I mean, even Jim Allison and Susan have shown that immunotherapy works so much more effectively if you do neoadjuvant trials. So what you can do in cancers like breast cancer, Laura Esserman is one of the big advocates of running early phase trials. There is called a window of opportunity between diagnosing a patient and doing a surgery. And during that time, you can test certain agents. Of course, you still have some safety study before, but that could give you an opportunity to test something in earlier stage, because metastatic stage, I'm not saying it's hopeless, and of course we want to make progress, but it's just so challenging. And I'm a big advocate of trying to move things earlier and testing them earlier, because that cancer, the way they evolve, you almost at the, something that's so resistant to everything, and you're not going to work with a single drug especially. And don't tell me how complicated it is to run combination trials, which took us 2 years to set up. So, but anyway, it's a very important point, and I think it needs to change the culture.
Yeah.
That protumorigenic program that I told you about, we have a window of opportunity study going in DCIS. And so women that are diagnosed with DCIS are randomized to this FDA-approved drug. We're just getting ready to break the blind to see if it helps. So I agree completely.